Today, we are very honored to have Ms. Cheryl Au, Senior Gemologist, Pearl, Gemological Institute of America, GIA, to share with us. Without further ado, may I now invite Ms. Au to come on stage. Ms. Au, please.
Thanks for the introduction, Adrian. So may I start now? Hello, everyone. I'm Cheryl from GIA. Today, I will be talking about pearls. I love pearls. They are such fascinating gems. They are mysterious, they are beautiful, that's for sure, and they are very complicated. We all love pearls, but what do we know about them? How much do we know about them? Today, I assure you that after the seminar, you will be able to understand the most important information about pearls. And it's even better if they come with GIA reports because you will know exactly where to look for this information. So before we get started, a little bit about GIA. GIA stands for Gemological Institute of America. It was established in 1931. Many of you know us as the creator of the 4Cs International Diamond Grading System. We are the foremost authority in gemology, including diamonds, color stones, and pearls. Some of you may not know that we are not privately owned. We are not publicly traded. We are an independent, nonprofit organization. All of our revenue is reinvested into research, academics, and to expand our mission. We are overseen by a diverse board of governors, including industry veterans and experts from research, academics, and businesses. GIA's mission is to ensure the public trust in gems and jewelry by upholding the highest standards of integrity, science, academics, and professionalism through lab services, research, instrument development, and education. In GIA, we have more than 70 years of experience in pearl identification and classification. I'm so very proud to be a part of this team because everybody in the team is just so passionate about one thing: pearl. So what are pearls? Pearl is a biogenic gem that forms inside the living body of a shelled mollusk. It consists of mainly calcium carbonate, usually in the form of aragonite, and a small amount of organic substances called conchyolin, and a small percentage of water. So when you're looking at a pearl, apart from only knowing that it's not an imitation and that it's a real pearl, what other information should you know or do you wish to know about that pearl? So today I've gathered 4 most important pieces of information that you should know when you're examining a pearl, which are identification, environment, mollusk, and treatment. So let's start with identification. So pearls, they can be classified into 2 main groups: whole pearls and blister pearls. And blister pearl is often confused with shell blister, and I'll talk more about that later. Well, to understand the formation of pearls, we have to understand the relationship between shells and pearls. The formation of both shells and pearls is controlled by the same type of cells. So they're called, can you see my, okay. They're called epithelial cells. So this type of cells, they secrete a calcium carbonate substance that's called mother of pearl or nacre. So let's look at the illustration there. You can see that's a shell layer, and under that we can see the epithelial cell layers. So these cells, they continue to secrete mother of pearl forming the shell layers, and under some accidental occurrences, these cells, they get transferred to the mantle. Maybe some damage is done to that layer, and then the cells get transferred to the mantle. And then they continue to form this pearl sac, this round pearl sac. And then they keep on secreting the mother of pearl, and eventually a pearl is formed. Well, a whole pearl is a pearl that stays inside the mollusk, inside the mantle tissue until it is found. So it's not attached to any part of the shell. Whereas blister pearl is a bit different. So blister pearl was once a whole pearl. But then let's look at that illustration there. Somehow it escaped from the mantle tissue, and then it got trapped between the mantle and the shell. Then you can imagine that the epithelial cells, they continue to secrete mother of pearl, covering both the pearl and the shell. And eventually the pearl will merge with the shell. So if the pearl is found at the very initial stage of attachment, that pearl may still be considered a whole pearl. But if that pearl has already completely merged with the shell, just like those images at the bottom, then those will be considered as blister pearls. And shell blister. If a shell blister is submitted to GIA, it will not get a pearl report, and it will only get an identification report. The reason why it doesn't get a pearl report is that it's not considered a pearl. That gray dot there, it doesn't represent a pearl. That's just a foreign material. It could be anything. It could be some parasitic worms or some marine organism. It's just somehow it got trapped between the mantle tissue and the shell. And then the epithelial cells, they keep on secreting the mother of pearl to cover this foreign material and the shell. So you can see in this process, there's no pearl that's ever been formed. And then at the end, there will be a lump, and that will be the shell blister. So that example there is a very interesting one because you can see the shape of a fish. So that's a poor fish that got trapped between the mantle and the shell, and eventually it became a shell blister. Okay, so when we are talking about identification, the main thing that we care about is whether the pearl is natural or cultured. So by definition, natural pearls, they're formed by various saltwater and freshwater mollusks within a natural pearl sac and without human intervention. Whereas cultured pearls, they formed within a cultured pearl sac with human intervention in the interior of productive living mollusks in a variety of conditions, depending on the mollusk and the goals. Some of you may recognize the necklace on the left. The drop-shaped pearl at the bottom, that one is a very famous natural drop-shaped pearl. It's one of the largest and most symmetrical pearls in natural drop-shaped pearls in the world. The name of that pearl is called La Peregrina pearl. So it's historically important. It was owned by royalty, and recently it belonged to the very famous Elizabeth Taylor. And I think currently that necklace is privately owned because in 2011, that necklace was sold at a Christie's auction for more than 11 million US dollar. So you can imagine natural pearls, they can be very valuable, especially when they are in good quality and they have this historical importance. And for cultured pearls, actually most of the pearls that we see in the market right now, more than 90% of them, they're cultured pearls. So the culturing of cultured pearls involves implantation of mantle tissues into the mollusk body. For bead cultured pearls, round bead made of freshwater shell is used as a nucleus. So the pearl sac forms around the bead and eventually forming bead cultured pearls. And for non-bead cultured pearls, as the name implies, there's no bead involved. So they usually come in different sizes and shapes. Most of the freshwater cultured pearls are non-bead cultured pearls, but recently we have seen more freshwater bead cultured pearls available in the market. And saltwater non-bead cultured pearls are usually byproducts of pearl farming, and they have this trade term called keshi. And you can find this trade term on GIA reports now. Right, so cultured shell blister, it's similar to natural shell blister. They are not considered as pearl. Somehow the farmer, normally they put a half dome-shaped nucleus on the shell right under the mantle, and then the mantle continue to secrete mother of pearl covering the nucleus, and then they cut that lump out, and that is the cultured shell blister. So Mabe pearl, many of you have heard of this term, Mabe pearl, is a trade name of assembled cultured shell blister. So a Mabe pearl does not form in the pearl sac as a whole pearl, so it is not considered to be a pearl. Basically, after the removal of the cultured shell blister, they remove the nucleus and they fill the cavity with resin or other filling material, and then they back the whole thing with polished shell for stability, and that's Mabe pearl. Those are some examples on the right and their products. So, to understand the identification of pearls, we have to study their internal structures. I know that many of you may just had lunch right before the seminar, so I'm going to ask you a question. So, can anyone tell me which pearl on this slide is a natural pearl? Anyone? Please? Yeah, that one. On the top left corner, that one is a natural pearl. So it's actually a half pearl. That's why we can see the cross-section through the setting. We can see some rings. We call that structure onion rings. It's a very typical structure of natural pearls. And you can think of that as some tree growth rings. So the inner rings are the older ones, the outer rings are the younger ones. And those two pictures on the right, they're actually the same pearl, just under different lighting. This one is a bead-cultured pearl. You can see there's a very clear round bead at the center showing the shell layers. And the pearl at the bottom, that one is a non-bead cultured pearl, showing a very typical elongated void at the center. Oh, and we don't always get to see cross-sections of pearls, but just for this show, we have prepared a very special pearl experience for you guys, and we have prepared some cross-section samples of pearls at GIA booth. So if you guys are interested to see more cross-sections of pearls, you're more than welcome to visit our booth later. So we don't always get to cut pearls, especially for client pearls, but we still have to study their internal structure in order to make some conclusions, right? So we have to use X-ray techniques. All of the pearls that are submitted to GIA, they go through real-time microradiography. So here are some examples of how the pearls look like under X-ray. So this one is a natural, this is a bead cultured, you can see a very clear bead at the center, and this is a non-bead cultured pearl. Sometimes RTX does not satisfy us because sometimes we want to see more clearly what the internal structure of the pearl looks like. So this is an example here. This is the pearl under RTX. We can see a shadow at the center, but we're not so sure what that thing is. But under X-ray computed microtomography, we can see very clearly that that's a foraminifera at the center. So this is another technique that we use to examine internal structure of pearls. Right, so you can find the identification result in the results section. Next one, it's environment. So pearls, they can form in both saltwater and freshwater mollusks. There are more variety of saltwater mollusks compared to freshwater mollusks that can produce pearls. I'm showing you a map, but I'm not going to dive into that too deep today. All I'm trying to tell you is that pearls, they don't just lie everywhere. You find certain type of pearls in certain area. And the reason why understanding the environment of the pearl is important is that, let me give you an example. Let's say you're looking for a South Sea pearl, and then some dealer approaches you and tells you that, oh, I've got a very nice freshwater South Sea pearl, and you instantly know that, oh, that's not right. The information that you receive may not be 100% correct because you know that South Sea pearls are only found in saltwater instead of freshwater. So knowing the environment of the pearl helps you to understand the background of the pearl you're looking at, and it helps you to make an informed purchase. So in GIA, we use different techniques to help us distinguish saltwater pearls from freshwater pearls. For example, EDXRF, which stands for Energy Dispersive X-ray Fluorescence Spectrometry. We know that this technique tells us the concentrations of some elements of interest. For example, we know that freshwater pearls, they contain a higher concentration of manganese. And knowing the concentration of manganese helps us to tell which pearl is freshwater. And the next one is X-ray fluorescence imaging. Because we know that freshwater pearls, they contain high level of manganese. That is also the reason why they fluoresce in strong green under X-ray. So that one is a nice example. The freshwater pearl there. Ooh, okay, that's the pearl that is fluorescing in green. That's a freshwater pearl. And the pearl below, I'm not sure if you can see that dot there. This is a saltwater bead culture pearl. The reason why we can see the dot is that that is the drill hole. So it's not the nacre that is fluorescing, it's the bead that is fluorescing, because bead cultured pearls are often, the bead inside the bead cultured pearls are often freshwater shell bead. And this technique is very handy when we're studying strands. Let's look at this strand as an example. You can see that most pearls in this strand, they are saltwater pearl, but there's one particular freshwater pearl here, and using this technique can help us to quickly locate that freshwater pearl. The next technique is LAICPMS. It is a powerful technique that tells us the concentrations of some elements of interest, also some trace elements. It gives us more information than just whether the pearl is a freshwater one or a saltwater one. For example, in 2019, we have published an article in Gems and Gemology Journal. We disclosed how we can use LAICPMS technique to discriminate the provenance of freshwater pearls, and this helps us to make conclusions on freshwater pearls with more confidence. And this technique is a quasi-destructive technique because it involves laser ablation, but then the destruction is very minimal. Let's look at the spots there. The laser spot, one laser spot measures 55 microns times 30 microns in depth. And the amount of nacre ablated is equivalent to 0.00007 carat per spot. So it's really minimal, and we can't really see that under naked eye. And it always takes some time for me to look for those spots, even under a microscope. So we can find this environment information right under identification row in the results section. Let's move on to mollusk. Well, these are all mollusks. Mollusk is a large group of animal, and not all of them can produce pearls, only the shelled ones, like bivalves, such as oysters, mussels, scallops, and gastropods, or univalves, including abalone, conch, and mallow. They can all produce pearls. So magnification is an important technique when it comes to gems identification. So for pearls, magnification helps us to study the surface structure of the pearls, which is directly related to the mollusk of the pearl. So there are two main groups of surface structures, which are nacreous and non-nacreous. So nacreous surfaces are formed by layers of aragonite, and those nacreous pearls are those that can exhibit this attractive pearly luster. And all the pearls that do not show this platy structure are non-nacreous pearls. Some of you may have heard of the term porcelaneous. So porcelaneous are those two pearls on the top here. So it's actually a subdivision of non-nacreous pearls. The reason why there's this name is that these type of pearls, they can exhibit this porcelain-like luster, and that they show this type of attractive flame structure. I'll show you more example later on. So here are some examples of mollusks that are capable of producing nacreous pearls. So note that all of the cultured pearls that we can find in the market, they are nacreous pearls. For example, Akoya pearls, they form in this Pinctada fucata martensi, and Tahitian pearls, they form in Pinctada macridifera. South Sea pearls, no matter if they're golden or silver or white or gray, they form in this species called Pinctada maxima. And all freshwater pearls, no matter if they're natural or cultured, they form in mollusk species that belong to the Unionidae family. So here are some examples. Okay, next one. Porcelaneous pearls, as I said, it's a subdivision of non-nacreous pearl. And here are some examples of porcelaneous pearls. Queen conch pearls and mallow pearls, they are some of the most valuable natural pearls in the market. You can always see them in auctions. Clam pearl. This clam pearl here shows a very nice flame structure that is characteristic of porcelaneous pearl. So here are some non-nacreous pearls. We can look at the scallop pearl in particular. You can see that the surface structure is interesting. It shows this patchwork cell pattern. I'm not sure if you remember, I said that pearl mainly consists of calcium carbonate in the form of aragonite. But for this particular pearl, it's form of calcium carbonate in the form of calcite instead of aragonite. That's why it shows this interesting feature. Right, so to make a conclusion on the mollusk ID, we have to have supporting data of various advanced techniques. But long-wave UV can give us some good indication. Using this strand as an example, this is a mixed strand containing pearls from different mollusks. But under long-wave UV, we can see that they fluoresce in different color. For example, those that fluoresce in red, orangey red, those are pearls from Pitheria species. Those that fluoresce in chalky yellow, those are from Pinidae family, and those that fluoresce in greenish yellow, they're from Pinctada species. So it gives us some good idea. All right, Oriental or Basra pearls. We always get asked about Basra pearls. What are they? So Basra is actually a city in Iraq that is historically known for natural pearl trading. Basra pearls are pearls that form within this mollusk species called Pinctada radiata, or the Gulf pearl oyster. So this type of oyster, they are local to the Arabian Gulf, also known as the Persian Gulf. So it is known for centuries for its rich and abundant pearl oyster beds, which made this area the natural pearl trade center, long before oil was discovered in the Middle East. And this picture on the right shows you the Pinctada radiata pearls. And it's very interesting that these kind of pearls are often displayed on a red cloth to showcase their nice color and luster. So this is a tradition that continues until today by most dealers in the Middle East and India. Right, so you can find the mollusk information also in the results section under environment. So let's move on to the last one, treatment. There are so many types of treatments that can be done to pearls, but I've only listed 4 of the most common ones. Modified color. So some color in pearls, they are more desirable than some other colors. So some people may choose to dye or process their pearls to make them look similar to those colors. And this is very easily detectable in lab. And for worked and heavily worked, because pearls, they form in nature, that's why they can have blemishes or they can be of shapes that are not desirable. So some people may choose to do some work on their pearl, for example, grinding the blemishes off or even to alter the shape of the pearls. So if this is found, we will say they're worked or even heavily worked if the working is very severe. And filling. So this pearl here is filled with metal beads. So the reason why this pearl is filled is probably because the person wants to increase the pearl weight, but there are some other reasons why other pearls can be filled. For example, some pearls can be hollow in nature, and as you can imagine, there's a cavity inside the pearl, so the pearl may not be very stable. So some people may fill some foreign material to fill the cavity to make the pearl more stable. That's one of the reasons. So the last one is coating. This is a very good example. You can see some coating coming off under a microscope. Yeah, this is also a very common treatment. So in lab, we use various type of spectroscopy techniques to help us identify the treatments. So all of the detectable treatments will be listed on GIA reports in the treatment section. Right, so I have covered all of the most important information that you should know about pearls. How about their quality? How do you evaluate the quality of your pearls? So in GIA, we use this classification system called the GIA 7 pearl value factors. So with no universal grading system for pearls in existence, the GIA 7 pearl value factors offer consistency and provide customers with the knowledge they need to make an informed purchase. So these factors are size, shape, color, luster, surface, nacre, and matching. I'm sure each one of you has this little leaflet on your hand, and you can find those information there. So you can find these classification details on our classification reports. Right, so our most classic pearl report is our pearl identification report, which details the quantity, weight, size, shape, color, overtone, identity, whether they're natural or cultured, mollusk, when it's determinable, environment, whether they're saltwater or freshwater, and any detectable treatments. Whereas this pearl identification and classification report includes all of the identification details, and it also includes the classifications for luster, surface, and matching. Right, so this is our newest report. This is mainly for saltwater cultured pearls. It's called Cultured Pearl Classification Report. It focuses on the GIA 7 pearl value factors, and it also comes with color treatment information. There are a few trade terms that can be seen on GIA reports. The first one is golden. So South Sea pearl oysters, they can produce different range of yellow pearls, and when they reach a certain saturation, they will be called as golden. Peacock is one of the most valuable and sought after colors in Tahitian pearls. And if the pearls, they have this dominant green hue and moderate to strong overtone, they will be called as peacock pearls. The last one is hanadama. So hanadama is a Japanese historical trade term that holds a lot of value in the Asian market, and it refers to the top quality of Akoya pearls. So these can all be found on our reports in the comment section. So some pearls or pearl pieces, they deserve special attention or special care. So we have some specialty services for those pearls. Notable letter, portrait report, and monograph report. Note that the notable letter and monograph reports, they are only by invitation. But if you think the pearl that you're submitting deserves some special attention, you can always request for a portrait report. Right, so I'd like to end today's seminar with the quote that I like very much. It's by Grace Kelly. The pearl is the queen of gems and the gem of queens. So I hope after the seminar today, you guys will be able to understand and appreciate this wonderful gift from nature from different angles. So I know we have covered quite a lot of information today. So as I said, we have created this very wonderful Pearl Experience Corner today at our booth. So this is our booth number. You're more than welcome to come and visit us, and you can also talk to me afterwards. Thanks again for the invitation by HKTDC, and thank you for attending this seminar. I hope to see you again.